everyone. Thank you for joining us today for Professor Sarah Richardson's talk on her recently published book, The Maternal Imprint, The Contested Science of Maternal-Fetal Effects. I'm Helen Zhao. I'm a PhD candidate in the Department of Philosophy and graduate fellow in Medical Humanities at the Society of Fellows Heyman Center. In a moment, Dr. Arden Hagley, who will be moderating our event today, will say a few words about the event and our sponsors. But first, I have the great pleasure of introducing our speakers. So I'd like to welcome first Professor Sarah Richardson, who is Professor of the History of Science and of Studies of Women, Gender, and Sexuality at Harvard University, historian and philosopher of science. Richardson is a leading scholar of gender and science whose work argues for conceptual rigor and social responsibility in scientific research on sex, gender, sexuality, and reproduction. She directs the Harvard Gender Sci Lab, a collaborative interdisciplinary research lab dedicated to generating concepts, methods, and theories for biomedical research on sex and gender. Richardson also serves on the standing committees for degrees in social studies and for the Mind, Brain, and Behavior Interfaculty Initiative at Harvard. And our respondents today will be Clare McCormack and Alexis Walker. Dr. Clare McCormack recently concluded a postdoc fellowship as a presidential scholar in society and neuroscience at Columbia University. And she's now joined the Department of Child and Adolescent Psychiatry at New York University Langone as a senior scientist. Her multidisciplinary research is focused on intergenerational transmission of adversity, as well as the psychological, neurobiological, and social processes that are critical for examining maternal child well-being in the perinatal period. Professor Alexis Walker is an assistant professor in the Department of Medical Humanities and Ethics in Columbia's Vagelos College of Physicians and Surgeons. She completed her PhD in Science and Technology Studies at Cornell, and her research investigates financial interests in biomedicine. And finally, last but not least, Dr. Arden Hagley will be moderating our event today. Dr. Hagley is a lecturer in the discipline of English and Comparative Literature and an affiliated faculty member with Medical Humanities and Medical Humanities and Ethics. She's interested in the intersection of medical knowledge with formalist and historicist literary approaches. Her research in Romanticism has been published in core journals such as European Romantic Review, Romanticism, the Byron Journal, and the Keats-Shelley Journal. And she is also published in the journals Partial Answers, Gender and Education, and Persuasions. She was a Society of Fellows Heyman Fellow from 2016 to 2019. So without further ado, I'll turn it over to Dr. Hagley.
Thank you very much, Helen, for introducing. So I'm Dr. Hagley, and Helen has already fully introduced my affiliations. I'm very happy to welcome Professor Sarah Richardson here tonight to present in our ongoing series, Explorations in the Medical Humanities. Her talk is supported by the Society of Fellows and Heyman Center for the Humanities and by the Institute for Comparative Literature and Society at Columbia. Many thanks to Helen, our graduate fellow in Medical Humanities, for organizing the event, and to Sarah Monks for providing technical support. This series, Explorations in the Medical Humanities, explores the enigma of how what we write relates back to the experience of bodies in different stages of health and disease. Our speakers consider how the medical and health humanities build on and revise earlier notions of the medical arts. This semester, the Exploration series is thrilled to host several book talks. I hope you also mark your calendars for the next one, which is Monday, April 25th, also at 6:15 p.m., when we will be hosting Professor Keith Wiley from Princeton to discuss his new book, Pushing Cool. Tonight's event is of note for the Motherhood and Technology group as well, at the Center for the Study of Social Difference. And it was through her guest presentation to the Motherhood and Technology group last fall on most sperm counts that I got to know Professor Richardson. We're very happy to welcome her back to Columbia to speak about the maternal imprint, which takes up the folk intuition that a woman may leave a biological trace on her gestating offspring. This is a landmark volume, and the implications of this research are far-reaching. from new ideas of post-genomic biology to transformations in prenatal healthcare to legal questions around surrogacy and the definitions of parenthood. I will turn it over now to the author and the respondents to open the conversation. Welcome, Professor Richardson.
Thank you very much for having me. It's an honor to be here and to be in dialogue with such an interdisciplinary group. And I want to thank the commentators, Clare McCormack and Alexis Walker, and the organizers, Arden Hegele, for the moderation, and Helen Zhao, who did not mention when introducing me that she is a valued member of the Gender Sci Lab and has been for many years, and a collaborator from whom I've learned a great deal. So let me begin. This is a talk about my book that is just out, and I want to start with this image. And this is the sort of image that characterizes the field of developmental origins of health and disease, or fetal origin science, that is the subject or jumping off point for the book. The DNA double helix is really today no longer the emblem of the leading edge of the life sciences. Instead, bodies, in particular maternal and fetal bodies, located in intergenerational time, in relation to social ecologies, typify the sorts of queries that are now at the center of post-genomic inquiry. These new biosocial sciences, such as epigenetics, hope to model at the level of biological mechanism how it is that bodies take up their environments with implications for health and life outcomes. In this book, I undertook to do a history of maternal effects science from the advent of the genetic age to today, analyzing three intertwined dimensions of scientific speculations about the long reach of the maternal intrauterine imprint. I'm interested in the draw of maternal effects science as a kind of disruption to determinist ideas about human fate. I'm interested also, secondly, in conceptual and empirical debate over how to study such effects given what I call their crypticity. And finally, I'm interested in claims about the implications of maternal intrauterine effects for women's well-being and autonomy. And in each historical period, scientists' views about what can be scientifically studied and indeed known about human maternal effects are entangled with cultural beliefs about men's and women's reproductive responsibilities, and shaped by scientists' politically and historically situated convictions about the relative importance of genes and social environment to human health and life outcomes. Now, these claims drew my attention at first because of some powerful narratives that were emerging from epigenetic science in the first decade of the 20th century. These claims include claims about the intergenerational inheritance of trauma, including these well-known claims about the intergenerational inheritance of Holocaust survivors. Rachel Yehuda, the neuroscientist who did some of these most prominent studies, is quoted in a 2016 United States Veterans Affairs press release as stating, "What happens to our parents, or perhaps even to our grandparents or previous generations, may help shape who we are on a fundamental molecular level that contributes to our behavior, or our behaviors, beliefs, strengths, and vulnerabilities." These claims have circulated widely. Natan Kellerman of the National Israeli Center for Psychosocial Support of Survivors of the Holocaust and the Second Generation, in a commentary on Yehuda's research, suggests that, quote, "the Holocaust left its visible and invisible marks not only on the survivors, but also on their children. Instead of numbers tattooed on their forearms, however, they may have been marked epigenetically with a chemical coating upon their chromosomes. Epigenetics, he imagines, might even provide verifiable data of trauma transmission that could carry legal consequences for claims of reparations." Beyond the realms of memory and history, the science of maternal-fetal effects, I have found, has become one of the fields of speculation through which we understand and process the risk and uncertainty of our present world. From the 2001 terrorist attack on the World Trade Center in New York City, which spawned a bevy of studies examining the effects of the event on children in utero, to our present COVID moment, where new cohorts of children exposed to the trauma of the pandemic are being assembled. Now, all of this research is part of an efflorescence in recent years of scientific interest in the long-term effects of the intrauterine environment. Beyond psychiatric disorders, researchers in a wide-ranging field of research on the fetal origins of health and life outcomes are searching for links between maternal factors such as diet, stress, and environmental exposures, and offspring outcomes such as obesity, heart disease, autism, asthma, even sexual orientation, attention deficit hyperactivity disorder, and other traits. While scientific interest in the imprint left by the womb has a long history, as I tell in the book, the proximate foundations of the modern day field of fetal origin science, which is sometimes called developmental origins of health and disease, or DOHAD, trace to the late 1980s. Using historical health records from the poorest areas of England, in 1989, British epidemiologist David Barker demonstrated that communities with the highest infant mortality rates had the highest heart disease mortality rates 50 years later. Barker argued that the risk of heart disease in these populations was linked to their earliest development in the womb, and therefore to the health of the mother, not only during that pregnancy, but throughout her life. Now, at the time, Barker's claims encountered disbelief and resistance, but today, this hypothesis drives multiple lines of inquiry at the intersection of developmental biology, teratology, nutrition science, epigenetics, and life course epidemiology. And as of 2014, more than 130,000 papers on fetal programming of disease were available in the biomedical research database PubMed. So in maternal-fetal epigenetic programming research, scientists cohere cryptic findings, that is, findings of small effect sizes with attenuated and cluttered causal chains between cause and effect, into bold, what I call biosocial theories, which locate causality and agency in the intrauterine period. Epigenetic explanations collapse scales of biological materiality from the molecular to the macrosomic. They collapse levels of biological and social analysis from metabolism to wartime trauma, and temporalities of memory, intergenerationality, and history into highly specific quantitative claims about levels of DNA methylation. And the result is really a compelling set of narratives charged with urgency, the specter of fetal harm, and with promise by researchers' pronouncements that a forthcoming applied science of fetal programming can redress it. Now, compared to scientific speculations on maternal intrauterine effects in previous eras, which I'll discuss in a moment, today's maternal fetal effects researchers benefit from a greatly expanded body of epidemiological data on the prenatal period. For example, beginning in the late 1980s, scientists initiated large-scale prospective cohort studies tracking mothers and their offspring from pregnancy onwards. The Avon Longitudinal Study of Parents and Children, or ALSPAC, at Bristol University in the United Kingdom, which began in 1991, enrolling about 14,000 pregnant women, is today among the largest studies attempting to track long-term effects of prenatal exposures and their interaction with genetics and other environmental exposures. ALSPAC has copious biological samples from its children and mothers, including maternal blood and urine during pregnancy, and cord blood and placental tissues at birth. And it has genetic samples for the mothers and offspring. And the study even, and it has data on children at 68 different time points, including the baby teeth of the children, which reside in the study's data bank collected by the study's appointed fairy. So this is a massive amount of data, and ALSPAC data yielded more than 700 scientific assessments of gene expression and epigenetic factors associated with things like obesity or asthma. But DOHAD science is not just mountains of data correlating natal exposures with later outcomes. Today's research combines this data with a set of guiding assumptions that serve as a conceptual framework for interpreting these data and launching research hypotheses. And these hypotheses are sometimes known as the mismatch hypothesis. DOHAD researchers believe that human developmental plasticity is greatest during the critical period of intrauterine growth, and that prenatal cues from the maternal environment can permanently program the developing fetus in ways that impact physiological functioning as an adult. So this is a programming hypothesis. The sensitivity of human fetuses to their maternal environment, they argue, is an evolutionary adaptation, allowing fetuses to attune themselves to their expected postnatal environment. So according to this adaptationist mismatch hypothesis, if metabolic and stress signals from the mother's body do not match the actual world the fetus encounters, as in the case of a nutritionally deprived fetus entering a calorie-rich American dietary landscape, illness results. And so Barker influentially argued that many so-called Western diseases of affluence, such as breast cancer and heart disease, were partly driven by a rapid lifestyle shift from relative deprivation in the early 20th century to the more hygienically and nutritionally transformed environment of the late 20th century. So, and because aspects of the intrauterine environment that a woman provides are themselves set by her own development in the womb, this mismatch between fetal programming and postnatal environment may, Barker claimed, take several generations to disappear. So many DOHAD researchers believe that in this way, intrauterine effects help explain how persistent social inequalities become embodied and pass across generations. That is, DOHAD research and maternal-fetal epigenetic programming research has become intimately linked to research programs interested in integrating the biological and the social, the biosocial. So University College London pediatrician and child nutrition expert Jonathan Wells, the author of The Metabolic Ghetto, 2018 book, argues that rising rates of metabolic disorders and obesity are so manifestations of the intergenerational transmission of health inequalities. Wells believes features of the mother's social and environmental context during her own development, including social class, may be transmitted to the growing fetus, conditioning it for a life of inequality even before birth. And these claims have been extended to explore, for example, the implications of intergenerational racism for things like heart disease rates. So the science of maternal effects, which is defined by evolutionary geneticists Jason Wolfe and Michael Wade as the causal influence of the maternal genotype or phenotype on the offspring phenotype, generally through the maternally provided environment, really offers a different picture of heredity than the one we learned in high school genetics. It suggests that mothers and fetus with more than just DNA. Infants in this worldview are programmed as they develop by maternal factors that influence the environment in which they grow. Research on maternal intrauterine effects in this way is thought to show just how profoundly our bodies are mediated by our environments, starting at conception. And applied to persistent patterns of social inequality, the phenomenon of intergenerational inheritance of trauma in human populations, maternal effects science offers, many believe, a powerful approach to understanding how our bodies are at once biological and social. So I wrote the Maternal Imprint to explore my ambivalence about these claims. Intuitively, I believe that bodies register their social and physical environments in ways subtle and profound, and that the maternal-fetal relation is a powerful and mysterious one. Today, maternal effects science is part of a broader and welcome turn away from gene-centric models of health, bodies, and biology. But as intellectually exciting and socially important as it is to appreciate bodies and biologies as shaped by their environments, the present intensive focus on a narrow window of human development, gestation, and on a particular class of bodies, women of reproductive age, requires scrutiny. So situating the intrauterine environment, critically determinative one for a range of life outcomes, articulates social alarm through women's reproductive bodies in ways that carry real implications for restrictions on women's reproductive autonomy. Moreover, even if such intrauterine effects exist, an exaggerated focus on the mother as the bearer of reproductive risk may misdirect resources from other important contributors to health and life outcomes. So I wrote this book to do several things. First, I'm really interested in the biosocial sciences. I'm interested in writing their histories, telling their histories. And because the maternal-fetal interface has been so entangled and so central with efforts to understand how the social gets under the skin, I saw it as one way to enter into and start telling that story, to situate and contextualize the recent rise in interest and investment in biosocial science in particular. My training as a philosopher of science, it always brings me to be interested in evaluating inferences, data and inferences in new scientific research on fetal epigenetic programming, in this and intergenerational inheritance of maternal effects in humans. I've been interested in the emergence of epigenetic science and the epistemic and methodological debates around the interpretability of that science when applied to human populations. And maternal-fetal epigenetic programming is one of the leading edge areas within that research. I, as a historian, knew that I would be able to show wide shifts in scientific and popular beliefs about maternal and paternal influences during the prenatal period over the past 150 years. The emergence of DOHAD research over the past decades is, some elements are new, but the ideas have a very long history, and our sense of what is common sense and where causality lies in parental effects research more broadly has varied quite considerably over different periods. I wanted to, of course, scrutinize the harsh mother-blaming discourses in the sciences of fetal origins of health disease within a frame of reproductive justice. And finally, I was interested in speaking to parents, clinicians, and policymakers to help them place in context the emerging scientific advice about maternal risks and to think about how or whether to act on that emerging science. So to walk through some of the episodes in the book quickly before we get to the comments. Of course, maternal effects, if we really start at the beginning, and here I'm starting in the late 19th century with the development of the germplasm theory of heredity, maternal effects contravene a founding premise of the modern theory of genetic inheritance, that is, that males and females contribute equally to the next generation. And I begin with this history, reconstructing what I call the principle of sex equality in heredity, as it was developed and really achieved by August Weismann, the architect of germplasm theory of heredity in the late 19th century. And I show how Weismann grappled with maternal impressions theories at the time as he developed his theory that the egg and sperm are isolated from the ravages endured by other bodily cells, and that the male and female gametes contribute equally, and how this was truly a radical break from previous reigning ideas about heredity, which in one way or another held that the mother and father contribute qualitatively and or quantitatively differently to the next generation. Now, what Weismann had to grapple with was the long history of maternal impressions theories, in part. This is one of the major asymmetric theories of maternal and paternal contributions to offspring. And maternal impressions refers to this notion that a mother's countenance, ambient environment, diet, fleeting impressions during pregnancy could imprint on her developing fetus, marking it for life and contributing to its future character. Maternal and paternal impressions theories asserted that heredity was not constituted solely by the transmission of material at the moment of conception. Rather, heredity encompassed the totality of characters shaped by the mother and father before birth. And both folk belief and leading scientists and physicians had long held that maternal and paternal contributions to the next generation were different. And Weissman challenged maternal impressions theories and some other asymmetric theories, such as spermic rejuvenescence and male and female complementarity in hereditary transmission. Now, these theories operated really at the charged interface between the natural and social order. They fueled intricate philosophies of what it is to be a male or female, what social roles men and women should have, and the responsibility of mothers and fathers for reproductive outcomes, and even the relative superiority of one sex or the other. And so we can see how Weissman's rebuking of parental impression theories really coolly set aside centuries of ideologically inflected assertions of the asymmetry of male and female roles in heredity by holding the contribution of male and female to be identical and presenting the refreshed idea that each generation begins anew and that beyond fulfilling basic nutritional needs, the maternal environment carries no particular consequences. Weissman's ideas, of course, engendered a backlash from those who felt strongly that our theories of heredity ought to encompass elements, an understanding of the environment's influence on intergenerational inheritance, and in particular, those who were interested in the special role of the mother in inheritance rose up in response to the increasing acceptance of Weissman's ideas. And I'm able to document in the next chapter of the book a sort of a revised maternal impressions theory that came up during the eugenics era under the term prenatal culture. So there was actually an upswing in maternal impressions theories after Weissman, and specifically in response to Weissman, rejecting Weissman's theory that genetic material passes from generation to generation untouched by its milieu. Prenatal culturists positioned the conditioning of the plastic fetus as a mode of controlling one's biological fate and overcoming a negative hereditary endowment. And against Weissman's dismissal of maternal impressions, the prenatal culturists offered a different stance towards scientific knowledge. They appealed to everyday experience, to common sense, and also to the likelihood that some realities may escape scientific probes and measures. Prenatal culture was, in the first instance, a vernacular scientific doctrine that offered a new version of maternal impressions theory suited to the eugenic age. So while today eugenics is often associated with hard hereditarian views of the sort that culminated in mass sterilization policies and so on in later decades, early 20th century eugenic thought embraced wide-ranging ideas about biomedical guidance of human advancement, and this included management of preconception health and the prenatal and postnatal infant environment. So many eugenics era authors and experts emphasize the importance of social and environmental reforms. And so the life of prenatal culture theories during this early eugenic period adds to our historical understanding, which continually bears repetition of the eugenic period as far from a period of reigning orthodoxies. And in the United States, prenatal culturists combined eugenic and progressive era ideas about social uplift through bodily conditioning with new theories about the critical importance of very early experience in brain development to press the importance of maternal behaviors during gestation. And intriguingly, they also proposed that the importance of prenatal culture should compel recognition of women's freedoms, including a right to an education, so they can maximally prepare her growing offspring for responsible citizenship. So prenatal culturists asserted a greater maternal part in heredity. Both in the law and the gospel of heredity, wrote physician and prenatal culturist Emma Drake in her popular 1901 advice manual for women, What a Young Wife Ought to Know. Both in the law and in the gospel of heredity of the two parents, the mother has a far greater influence, we believe firmly. And here is physician and eugenicist Mary Melendi in her 1903 prenatal culture text, Womanhood for Maidens, Wives, and Mothers. Both in body and mind, the mother's powers of transmission of habits, good or bad, are many times stronger than those of the father. And here is the wonderful Georgiana Creedy, a California suffragist and feminist, who wrote in her book Transmission, or Very Character Through the Mother, that as society comes to recognize a woman's great maternal power in the transmission of traits, that, quote, her character will broaden, her thoughts enlarge, and subserviency will no more belong to her than her brother. So presenting the space between a pregnant woman and her growing fetus as an inscriptive, socially imbued environment with implications for the adult offspring and for future generations, maternal impressions theories reveal deep entanglements with reproductive and gender politics. And this terrain has been well examined by historians and gender scholars. So on the one hand, the theory of maternal impressions appears to attribute a greater power and agency to the maternal body in reproduction and heredity than has historically been the case. But this agential reading of maternal impressions is complicated by the theory's conception of the maternal body as irrational and indeed more of a passive conveyor than an essential shaper of fates. And many historians have shown how the theory of maternal impressions was instrumental in creating, stifling imperatives, and even pressing advice on women. Right. So we now shift to the scientific eugenicists who were in a rage about the prenatal culturists and responded by batting it away as old wives' tales, as superstition, and absolutely not science. And they were quite successful in this. By 1920, prenatal culture theories were firmly rejected by the mainstream scientific and medical establishment, really completing the rupture from previous beliefs about the role of the womb in heredity initiated by Weissman. Biological scientists and medical doctors now dismissed any notion that the mother, except in cases of extreme deprivation or injury, could alter her offspring's traits. And so in the Journal of Heredity in 1914, there's an excoriation of prenatal culturists. What can parents do to determine before birth the character of the child? Nothing, except so to live that the germ cells are not enfeebled. And here's the first manual on prenatal culture from the US Children's Bureau in 1913, averring that there is no connection between the mother and the child in the uterus. During this time period, eugenic discourses focused on the germplasm, the health of the egg and the sperm, and way over and above the intrauterine period. And this led to the surprising reversal, really, toward a focus on men's reproductive health and men's responsibilities. So because adherents of Weissman's germplasm theory of heredity conceptualized prenatal health as the proper care and hygiene of the sperm and egg, interest in parental reproductive responsibility and early developmental exposures focused on both parents and on the period prior to conception. And at the same time, prevailing ideas held that environmental risks to the human gene pool were most likely introduced by men with their riskier lifestyles, occupations, and consumption patterns. So thus, in contrast to the prenatal culturists and really to doughhead scientists today, scientists in the 1920s and 30s focused their prenatal advice far more on the preconception period and on men than on intrauterine exposures and women. So men were exhorted to protect their sperm by abstaining from alcohol, undergoing regular syphilis testing and treatment, and maintaining their bodies in overall good health. And women, on the other hand, were instructed to evaluate their potential partners by the avidness of their healthy sperm producing lifestyles. So reflecting on this period throws into dramatic relief the peculiarity of today's view that a focus on maternal pregnancy effects in producing offspring outcomes is just common sense. So how did we get from there to here? During the same decades that human geneticists and medical doctors were waving away the possibility of maternal intrauterine effects on heredity and development, anomalies to Mendelian patterns of inheritance began to accumulate in the scientific literature in plant and animal genetics. For some traits, it appeared that maternal phenotype was always determinative, regardless of paternal contribution. And scientists wrestled with how to interpret these findings, eventually agreeing on the ambiguous term maternal effects to refer to a wide class of observations of maternal phenotypic influence on the next generation in the absence of genetic transmission. In applied contexts, such as breeding agricultural strains of plants and livestock for desired qualities, or the mass production of cloned mice for use in scientific research, and the development of in vitro fertilization technologies for the treatment of human infertility, maternal intrauterine effects became not merely a matter of speculation or an intriguing anomaly to Mendelian theory, but a force that had to be reckoned with to achieve human aims. And so I tell the story in the book of how in the 1940s and 50s, new techniques in reproductive biology allowed maternal effects to be studied experimentally in non-human placental mammals. And these experiments established that maternal effects, while cryptic, can alter heritability estimates by changing, for example, rates of fetal growth. So evidence that the intrauterine environment could matter, particularly for macrosomatic traits like body size, led some to wonder whether maternal effects might be at work in human health. In the next chapter, I then look at the long history of debates over the origins of the birth weight gap between African American and white American babies in the United States. So it was in the context of this long history where for many decades, the idea had predominated that the gap in birth weight, so on average, Black Americans are born smaller than white Americans. And that gap, although both groups have their averages have gone up over time, the gap still persists. That gap had long been thought to be genetic in origin. And it's in the 50s with the examination of large bodies of data on birth weight that in relation to social variables, that this hypothesis is reassessed and the theories of maternal effects begin to enter biosocial theories of racial differences in health outcomes. So that is the origins of the birth weight disparity and its implications for health and social inequalities across lifespan has long been a matter of debate among biologists, epidemiologists, and social scientists. And I tell that history in this chapter. And now birth weight itself began to be regularly collected as a demographic variable beginning in the 1950s. And that's when researchers seized upon it as a possibly rich variable reflecting on fetal well-being. And then scientists, including some underdocumented scientists at predominantly Black medical institutions, drew on maternal effects theories to forge the hypothesis that racial differences in birth weight demonstrate that racial discrimination and deprivations can leave a mark even before birth, and that the intrauterine environment might serve as a mechanism for amplifying these effects across generations. And these maternal fetal theories of racial variation in birth weight even entered 1960s and 1970s debates over whether racial differences in educational attainment and IQ are due to genetic or social factors. So debates over racial differences in birth weight from the 1950s to the 1970s raised the prospect of mining the prenatal period for cryptic as yet acknowledged drivers of health and social inequalities. And this vision of prenatal growth metrics as ripe material for formulating biosocial hypotheses about how inequality gets under the skin linked maternal effects science to this really dramatic picture of present day human bodies as dialectically constructed by past environments of their ancestors, as products of power structures and politics, and as tools or vectors or instruments through which we can today change relations of power. And appreciating this entanglement of maternal effects science with the project of documenting and ameliorating the biological dimensions of social inequalities is critically important to understanding where the field is today. All right, I'm coming to a conclusion here, and then we'll go to the commentaries. So today, scientists view birth weight as too crude a measure of fetal outcomes to support causal hypotheses about maternal pregnancy effects and later outcomes. Instead, maternal fetal effects researchers have turned their attention to the new science of epigenetics, which refers to molecular changes in the non-DNA regulatory apparatus of the genome. And I love this cartoon by my colleague, Andrea Beccarelli, who, this is typical for the field. You have the arrows into the very pregnant abdomen with the almost infant-sized child, headless pregnant abdomen, arrows out, leading all eventually to an ambulance rushing the child off to the emergency room. So epigenetic markers such as methylation can change in response to environmental stimuli. And if these changes can be shown to be induced by the intrauterine environment, remain stable over time, and have functional implications for human health and biology, epigenetics, it is believed, may offer a mechanism for the long-term effects of maternal pregnancy exposures. And this is really a post-genomic science exploding in the years since the conclusion of the major genome sequencing projects around 2010, offering the prospect of understanding the genome as a dynamic reactive system integrating environmental inputs. And all of this has led to great excitement about epigenetic science across many fields of the life and social sciences. In the book, in a detailed analysis over 2 chapters of studies in this field, I show that there exist large methodological and conceptual obstacles to establishing epigenetic mechanisms as stable, informative markers of maternal intrauterine exposures and as a specific cause of later outcomes. And I argue that the level of interest and excitement about maternal fetal epigenetic programming far outstrips what the science actually can and does show. Our intuitions about the strength and plausibility of causal claims in the area of maternal fetal effects science are, I argue, profoundly shaped by deeply carved assumptions about where agency and responsibility lie for offspring outcomes. Now, today, this work, some of it done in non-human organisms, is part of what Dorothy Roberts has called a new biosocial science, explicitly positioned as a progressive democratic science that is aiming to help policymakers confront the reality of social inequality. And yet at the same time, positioning, recreating categories of good and bad parenting, and suggesting self-perpetuating biological lesions or deficits in certain populations exposed to bad mothers. Although the search for, a progressive biosocial science at the maternal-fetal interface, of course, has earlier roots. As I conclude, I want to look at sociologist Dalton Conley's 2003 book, The Starting Gate, Birth Weight and Life Chances. And Conley here outlined a biosocial science that would focus on the well-being of children and mothers and use scientific knowledge to help illuminate and redress the harms of poverty and racism. And now today, this vision is widely shared. There's a growing body of human epigenetics findings reporting correlations between adverse early developmental effects, biological variables such as methylation levels, and health outcomes or life outcomes. And there are several funding initiatives that are responding to this flowering of biosocial science, integrating biological and social science approaches. And you see very strong claims about the integration of biological and social science perspectives, sort of imagining an ambition for conceptual and disciplinary unification through a sort of super science that integrates the biological and social sciences to frame and explain fundamental questions about our collective human experience. And here I'm quoting Michael Meany and Moishe Ziff of McGill. Now, the problem is the disjoint between social categories and what biological measures actually capture. The push for this biosocial science glosses serious conceptual, empirical, and methodological questions. In the excitement over epigenetics, are we attending to the way in which the biological, and particularly the molecular and genetic, is privileged with a special explanatory authority as a measure of concrete realities? In the case of maternal effects science, what is at stake for our picture of the social dimensions of health in asserting that methylation levels represent a valid and generalizable measure of social processes and structures as experienced by diverse individuals and populations in the womb? As Dorothy Roberts puts it, to fit social facts into a biological framework, some scientists convert social inequality into a series of deleterious environmental exposures and developmental deficiencies. In maternal-fetal programming science, that environment is a human woman, and the deficiencies a direct result of her cues to the fetus during prenatal development. So here the social order fades from view, and attention is focused on the biochemical processes within the body, rather than inequitable social structures, right? And then these biochemical responses are seen as programmed, as permanent, as even heritable. And the result is a picture of fetuses with what Roberts calls self-perpetuating biological deficits. So the outcome is really to obscure the role of social factors and the ongoing effects of ongoing disadvantage. So I've argued that epigenetic research on maternal effects advances a model of human inheritance and development in which the wider social and physical environment can be seen as heritable and determinant of future biomedical outcomes via discrete biochemical modifications introduced by the amplifying vector of the maternal body. So rather than challenging genetic determinism, as biosocial discourses long hope to do, and rather than challenging biological reductionism, these present-day research programs collapse the distinction between determinist and biosocial or anti-genetic determinants approaches. Instead, they strategically appropriate and modify these discourses to include a particular conception of the social determinants of health, one that places the maternal-infant relation at the center. And I like to close with this image, right, of from the cover of Der Spiegel, just an article on epigenetics, and you can see, you know, the intensity of a particular conception of the body, that is a feminized body, that is these issues of gender, heredity, and the post-genomic moment, I think, are powerfully integrated in this cover image chosen by Der Spiegel. You get a nude, white, blonde, model gorgeous woman emerging from the water, curling around her body in the shape of a DNA helix, and in the background glow DNA sequences reading out. And shaking off her double helix, the woman rises above her gene pool. Clean, pure, and born again, she gestures upward toward the heavens, above the muck of the pond and her genetic readout. And bold letters read, "Victory over genes, smarter, healthier, happier, how we can outsmart our genetic material." And it's conveying the promise of epigenetic science for health optimization and shaping human destiny, while at the same time packaging a dense cluster of feminized associations, from birth, fetus in the womb, to ghostly apparitions, to the prolific symbolism of erotic nudes, and the forward-looking guardians of the future family in socialist and pronatalist propagandisters, and conventional representations of cleanliness, purity, and beautification in cosmetics and naturopathic advertising. And we see that epigenetics does not so much make plausible this biosocial embedded body, but rather it fixes the molecular gaze upon the embedded body, an already formed and highly charged entity in the science of maternal-fetal relations, and elevates it to the center of biomedical theory. All right, I'll conclude there. Thank you so much, and I'm very much looking forward to the comments.
Thank you so much, Sarah. So we will hear the first set of comments from Claire. Go ahead.
Thank you. Thank you for inviting me to be part of this conversation, and really thank you, Professor Richardson, for writing this book. It's totally original, it's sharp, it's an amazing read, synthesizing just a huge amount of research in historical and contemporary science, and as well as being accessible to a wide audience. I think it's really essential reading for my colleagues working on fetal programming research. I would have loved to have read this in grad school. And somehow I think it's easier for us to recognize that science is always entangled with cultural beliefs when we look backwards. But the journey that you took us on in this book prompts us to be critical of the approach and the claims of contemporary maternal effects research too. And so I just thought I would reflect on some of the takeaways for me of reading this book as a scientist. So I've worked on studies looking at how different factors in the prenatal and the postnatal environment confer risks for offspring in terms of cognitive development. First, I began with alcohol exposure, and now more recently moving to look at stress, depression, and trauma as exposures. And I don't think the research into the effects of the prenatal environment on child outcomes will be slowing down anytime soon. So the question is, how can we do better and more careful, thoughtful, impactful work? So firstly, I think one of the clear takeaways for me is that the questions we ask really matters. And if we don't give sufficient context to our findings about certain maternal attributes or behaviors being risky for the fetus, I think you warn in the book that this science could just function as another stigmatizing, deterministic discourse, contributing to stigma and moral panic. And I take this warning very seriously. Sometimes alarm bells do go off when I see an article with a title or a conclusion, especially, that could feed straight into that narrative. And as a peer reviewer for journals, I probably annoy authors about fussiness over their word choices in titles and abstracts especially. But I will say that in my experience doing the work is that I've always worked with colleagues who are themselves the first to agree that social inequality really is the problem, not women's behaviours or bodies. So you'll hear researchers say, well, of course we can't fix poverty or trauma or discrimination, but what I can do is look 2 levels down at what we might call modifiable risk factors like stress levels, diet, pollution, drug use, social support. And so the rationale of this solutions-oriented science directs us to look at these things, because if we see effects there, some change might be actionable now in this generation for a woman who's pregnant today. And I think it's also clearly something that women want. You only need to look at the popularity of books by people like Emily Oster, the advice mill of the internet, to know that pregnant women really do want this information to make decisions. And increasingly, they want to see the actual data to back up the advice that they're given, which is great. So we're speaking to a really motivated audience that want to know if something they do will help mitigate, effects of their adversity, or even just to see if they can optimize their behavior or the environment to give their kids one more IQ point. But what this book really has laid bare very plainly is that although this research may be done in good faith by well-meaning scientists, there of course can be unintended consequences. It's very hard to publish a study showing a link between maternal behaviour and kid outcomes while also dodging mother blame. Unfortunately, very real guilt, worry, and mothers blaming themselves for any problems they might see in their kids is what happens when data on the mother and the uterus is just about the only data they see. And that this discourse is internalized. So maybe we need more creativity and to take more responsibility in choosing the questions that we ask with research. One brilliant study that comes to mind, and I have to mention, is recently published data from a study led by Kimberly Noble at Teachers College. This was a randomized control trial looking to improve child brain and cognitive development in a really diverse sample of low-income families. And the intervention was totally simple. It was just cash. It was an unconditional monthly cash payment of around $300 given to mothers with newborns across the child's first year. And then, yeah, the results that were just published really recently showed the intervention led to significant differences in brain development for kids whose mums received the cash, showed more high frequency brain activity compared to the low cash group, associated with, better development in thinking and learning. So that study leveraged the very potent idea of developmental origins of health and disease that catches on, but avoided mother blame by kind of judiciously choosing an intervention that actively empowers mums. And the implications of that one were really crystal clear, ready to translate into policy whenever, ready. And I guess this was, to me, a case of maternal effect science, leveraging its own power and impact, but aiming the intervention target not at mothers and their bodies, but higher up at some of these external forces. So my colleagues and I have a similar motivation in research on maternal mental health, looking at stress and depression during pregnancy as fetal exposures. This, of course, faces the problem that you describe of cryptic effects in the book, so that, we're often talking about statistically small effect sizes that may or may not be of immediate clinical significance. And also, sometimes the effects of prenatal exposures are measured later in kids, although not always. Sometimes you see effects in the brain and behaviour of the fetus and the infant during pregnancy or soon after birth, taking away the issue of postnatal confounding. But still, I don't personally need to see any effects on kids to be concerned about mothers' happiness and well-being in and of itself, just like I don't need to look at kids' brains to decide if we should give their mum some financial support. But I think it would be naive to ignore the power of these studies showing how these things do affect the next generation, and that it potentially could spur more action. Maybe I'm optimistic, but if the end result was more investment in women's mental health, more support for mums, I can feel more okay about doing this so-called, prenatal programming research. And I think while this search for mechanisms of exactly how these experiences get under the skin continues, and it should, there's plenty of evidence already to kind of spur this action as it is. Also, I think it's a reminder just for researchers to set our standards very high in terms of statistical analyses and being very clear in explaining the clinical significance of findings, not just statistical significance. Also a reminder about replication, that we need to, replicate studies in multiple populations before we get too excited about findings. And maybe most importantly, to make sure that conclusions line up with the data, especially because this fetal programming research is totally irresistible to the media, as we saw with Rachel Yehuda's work and on intergenerational trauma and others. Another major takeaway for me was facing up to the kind of inexcusable at this point paucity of research on fathers and partners. I think you had the figure that there were 20 times the number of studies on mums as opposed to dads in the DOHAD field, which is really alarming. And so clearly, we can do better. It's easier said than done, but I think we need to be more creative and deliberate in bringing dads into research. And finally, there's a call towards the end of the book, and as you said today, to think about a future in which prenatal and periconceptional exposures of all sorts, maternal, paternal, and other relevant environments, are explored in tandem and weighted in context. And I do see some hope here with some of the emerging national big cohort studies, and also with the push towards open science and data-driven approaches. And specifically, I'm thinking about the 3 huge NIH initiatives. I think you used the word super science, talking about some of these. So there's ECHO, which is the environmental influences on child health outcomes, the ABCD study focusing on adolescents, and then there's another one starting now, the HBCD study, the Healthy Brain and Child Development Study. So the idea is that dozens of sites around the country collect data on a huge range of brain, behavioural, biological, environmental information. They follow up kids over time. And then using open science frameworks, there's an opportunity for that data to be analysed by anyone. there's an endless number of questions to be asked from the data. And you can compare studies from the same data set to see how the framing of questions leads to totally different conclusions sometimes. One example from ABCD study, the adolescent study, one paper was showing that of maternal alcohol use during pregnancy on behavioral problems in adolescents. And the conclusion was that people should avoid drinking when they're pregnant. But then another study used data from the same children at the same from the same data set, but they included dozens of environmental factors, and they used a data-driven approach to ask the question of what factors matter in predicting adolescent problems. And they found that when you consider all the other factors, alcohol use was actually not a significant predictor. By a huge margin, the biggest predictors were parent psychopathology, the school environment, and the neighborhood. Same study, same data, two different conclusions, but you can guess which one made media headlines. And so I just thank you again for the opportunity to be involved. I need to stop now and pass it over, but so much to say. Thank you.
Thank you so much. And we'll now hear Alexis Walker's comments. Please go ahead.
Hi, folks. I'm so glad to be here with you all. Dr. Richardson's book felt really quite personal to me in many ways, and honestly, I suspect that'll be true for a lot of people because the book is so expansive in both its breadth and depth, which is of course quite a feat. And touches on so much in American history and history of science, et cetera, from the late 19th century to the present. So, we've got this great analysis of early 20th century science fiction with a really nice discussion of Aldous Huxley's 1932 Brave New World. That's in chapter 4, where Dr. Richardson so nicely points out that in that work, the production of people for a specific social function, Huxley imagined as the result of manipulation of the environment of eggs, sperm, and embryos, right? Not of nuclear genetic material, but these environmental factors. And I think that was such an interesting reading. And then, we fast forward to chapter 7 of Dr. Richardson's book. We've got, among other forms of evidence, that Dr. Richardson marshals evidence from a Twitter discussion on mediation analysis and epigenetics research. So really an extensive set of sources, time periods, and locations here. So, I mentioned American history, but of course the focus of the book geographically is broader than that. And I'd love to hear Dr. Richardson speak a bit about the book's nature as what I read as a US-focused investigation in global perspective, perhaps mainly Euro-American, over what is quite a large time span. That does seem to be more and more rare in the literature, although I'm not as up to date on history of science circles in the last years, but I'm curious about your thoughts on the boundaries of the study in terms of time and space. So, this book about maternal-fetal effects, took me back to the plant biology research that I conducted at the Max Planck Institute in Cologne right after college. And I'm just going to mention a few little personal connections because I think it speaks a little bit to the way that the sort of far-reaching elements of the book. So there's plant biology in here. Of course, there's I had based on that a lot of discussions with my dissertation committee member Simon Seth, who's a great historian of science, on ideas of race and heritability in plant sciences, animal sciences, and human sciences. And Dr. Richardson does such a nice job of bringing those together in the book. So in chapter 5, when she's looking at the animal and agricultural sciences, we saw the picture of the pig, and mentioned, the book you mentioned the swine geneticist Gordon Dickerson in Ames, Iowa. That was, I saw that on your slide, where my great grandfather was a civil engineer, and I imagine him knowing them knowing each other in this small community of engineers and scientists in Ames. But in any case, these connections to so many parts of American history, and then also, and I'm again on my other side, in your discussion, you mentioned briefly in your talk just now of Black physician researchers involved in highlighting the social elements of racial birth weight gaps in chapter 6, that really took me to my father's experience as a Black physician in the United States. For example, you cite Edward Perry Crump's 1959 piece highlighting anti-Black discrimination in medical training, and my father started medical school 6 years later. So anyway, so lots of personal connections in just a way that brings together such wide spanning intellectual and social history, which I just loved about the book. And then that is then met with a really deep analysis of the science at hand. You know, I had a little heart in the margin. There is a quoted, I took it here on page 171, where Sarah notes, quote, "The following section may be hard going at first for many non-scientists, but understanding how scientists conceptualize and study methylation variation in the human genome will prove worthwhile once we get deeply into the landscape of current research in this field," end quote. And that is certainly true. And this is not the first place in the book where we start getting into some weeds of the science, but Sarah writes so in such a fashion that keeps that really exciting throughout the book. And there's also so many great characters, right? So coiling snails and Shetland horses, etc. All sorts of fun things to be reading about throughout the book, especially when you pair it with such interesting analysis of things like the philosophical and social nature of causality, which I'll talk about more in a minute. I really loved. But in chapter 7 on fetal programming, it is a place where you really dig into the science in order to refute many of the claims being made by today's epigenetics research, which is that's a load to bear for a social scientist. And I'm curious a little bit in terms of, you do a few places rely on the arguments that have been made by bioscientists, but I'm curious a little bit to hear about responses to that. It was very nice to hear Claire's enthusiasm, but I do wonder how fetal programming researchers have responded. I know you've published in Journal of Developmental Origins of Health and Disease. I saw that cover also in your slides, and so I assume that's a way of speaking to that scientific audience. But I'm curious about sort of both your strategies and how that's been received. And because I think, you do such a nice job of pulling out looking at things like levels of effects that are seen as acceptable in human versus animal research, right? Lower bar of evidence looking like in human research, and looking at studies for lack of attention to tissue specificity in methylation studies, and the problems of reverse causation in terms of that epigenetic markers, of course, could be produced as a result of certain phenotypic variation as opposed to causing that, and so much into the sort of meat of the science, which raises questions about our authority, I think, of where we as social scientists get to speak, and humanists, perhaps, I have to say. It's also a place where I thought quite a bit about how our disciplinary and methodological approaches shape the conclusions that we as social scientists and humanists reach. So as an ethnographer, I find that sometimes it's difficult to critique the actors that I study. So in my doctoral dissertation work, I looked at international financial institutions' work in health programs, their health loans, et cetera. And I thought I was going to write a much more scathing analysis than I did in the end. And, I don't really want to, it's not to talk just about going native, but I do think that, I'm curious to hear about your relationship to your contemporary actors, and how maybe studying them as a historian and philosopher may be different than folks who have been studying epigenetics from a sociological interview-based research. I have a few more quick little things. I know we want to leave time for a little bit more discussion. I also really thought, as somebody who has a fair amount of scientific training myself, I thought that that fetal programming research was much more settled than it appears from the book. So I think that's really, I thought it was wonderful to see that come out here. And then about the social nature of proof and causality, just so helpful for thinking about so many parts of science, not just fetal programming. So the idea that, lower evidence bars are needed for scientific claims that confirm our social ideas, I think is just such an eloquent way of putting together so much of social science of medicine, or of social studies of medicine and science, right? So, but it also has a unique position, as you described, in contemporary, the contemporary context. So I do think that, certainly it's true of, 19th century racial science that people have not necessarily set out to prove XYZ, but rather they already know that thing, and therefore are unsurprised when they find it. But I think, of course, how you pull out the unique nature of probability and sort of cryptic effects makes that challenge all the more clear in 21st century science. So I think that is just really, for me, it was the way it's phrased is helpful for thinking longer term about history of science and medicine, but also for the specifics of the more recent period. So I had some more things that I want to talk about, but I think in the interest of time, I will leave it there. And if we have time, we'll come back. Thanks so much.
Thank you both for fantastic comments. Sarah, we will give the final words to you to respond to the comments.
Thank you so much. And if there are comments in the chat or whatever, I hope you'll send them my way and I'll respond as I'm able. But I just want to thank both of you for these incredible commentaries on the book and your reads. It's amazing to be in dialogue with you. Quickly, Alexis, thank you so much for appreciating chapter 5 on the plants and the animal research. There's a particular kind of reader who loves that kind of history of science, history of biology, and it was real hard to write. So thank you. And your family connections to these different moments in American scientific and medical history. Yeah, in a way, through this history of maternal-fetal effects, you land at a lot of the sites of the history of biology in the 20th century. And it might cause us to question as historians how much we ourselves have centered. We've been critical of the centering of the gene, right, in the sciences. And in a way, our histories and our social studies have replicated that. There are whole other histories, like what if we move the axis of action and realize that the sciences of pregnancy and reproduction have also been central sites of knowledge production and have their own stories and nodes of power. Yes, both of you in a way, Alexis, with your question about responses to my analysis of epigenetics, and Claire with your deep and probing questions about how researchers in your field can take some of this on and do science that is reflective and accountable to these kinds of questions. Yeah, I mean, I think that so first of all, as to responses to chapter 7, I've had a lot of positive responses. In fact, I'm really not saying much new in the broad generalities. Some of the specific critiques of these studies do come out of my close reading in tandem with, talking with researchers in the field and having others read it. I do a close read in that chapter of 3 major lines of research. Usually those are like citations 1, 2, and 3 in any critical review of human epigenetics work. Maternal-fetal epigenetics work in particular. And that is the Dutch Hunger Winter Studies, Rachel Yehuda's Holocaust Trauma Studies, and I do Suzanne King studies on experiences of stress during trauma or during pregnancy, which is sort of part of that larger milieu of important work out of Montreal in the field. And I haven't had responses from the authors of those studies so much, but I think that what I am trying to do from a history and philosophy of science perspective there is extract and understand the conventions and norms of reasoning in the field. It might read in places like scathing criticism, but really my goal is to say, all right, what's the evidence? What are the claims? How do they fit together? And to align them in in an evaluation of how the field moves forward and how it works to replicate or not its findings. So I see it more in the spirit of characterizing and very much continuous with questions that practitioners in the field, especially epistemically sophisticated ones, are asking themselves. And so I'm quite aware that scientists are concerned about these issues of cryptic causality, what that I call cryptic causality, and trying to triangulate and move around it. My argument, however, is that we should prepare for the likelihood that this problem of cryptic causality is actually characteristic of the field and will be persistent. As for Claire's excellent questions about how the field can do better, and the wonderful examples of Claire's own research as well as Kimberly Noble's research, I think these and the example of with bigger cohorts being able to ask different questions using different frameworks of the same data, which I think can be very productive for honing and analyzing assumptions. These seem like promising developments to me. I do question, though, I really empirically question the idea that we must study these maternal-fetal effects because it is a particularly effective way of bringing resources to mothers. And that claim is made a lot by folks in the field because, I mean, absolutely, people in DOHAD and people in maternal fetal research in general, they are progressive, democratic, biosocial scientists who want to bring resources to mothers. That is absolutely the case. And they truly do believe that this is true. That if you do more of this kind of research, the funding flows. That's an empirical question. I wonder if it's true. We believe that if we healthize it, if we biologize it, if we show it affects kids, that the resources will flow. And I think that's something we need to probe with specific case studies and so on. But nonetheless, I appreciate the many attempts that are going on in the field, and I know some others as well, to really engage and carry out reflective research that engages the mothers themselves and fathers, and it takes into account many intervening and interacting social factors. And so I agree with you. The field agrees that social inequality is the problem, right? And so then the question is, what kind of work is this science doing in the world? And I'm so interested in the possibility in this post-genomic age with these cryptic effects and these data-driven sciences, if we can develop a way of thinking and talking about science that dwells in and explains the nature of uncertainty and contextualizes risk in these fields and as it translates the science, right? And instead, it seems we're often stuck in a hype cycle necessary for funding and for public translation of research. And I'm so interested in building a different way of doing and thinking and talking about these pressing areas of the sciences that are moving to the center of and really are a model for post-genomic biomedical research. So thank you for this wonderful conversation. And I'm sorry we don't have more time to talk, but hopefully we can follow up in other ways.
Right. Well, I think at that point we are at time. I would like to thank everybody for participating. Professor Sarah Richardson, Dr. Clare McCormack, Dr. Alexis Walker. Thank you for fabulous presentations, responses, and thank you everybody who attended. If you have lingering questions, please feel free to get in touch with me or with any member of the panel with your questions, and we will make sure that they reach the right destination. All right, have a safe and peaceful night, everyone, and thank you again for attending.
Thank you.
Thanks so much, folks.